Okay, so my name is Bria Moore,
I'm Keith Johnson.
Naisha Paul.
Willie Cornegay.
and we will be discussing cell motility. To begin, we're going to talk about the cytoskeleton. The cytoskeleton is a network of fibers that extends through the cytoplasm. It's made of protein filaments and tubules, and it's in the cytoplasm of many living cells. It gives this cell shape and coherence, and it organizes the structures and activities of the cell. Um, it's very organized, as you can see in the figure. Wait, the figure here. The role of the cytoskeleton is to provide structural support and help maintain cell shape. Um, as the cells move around, they tend to uh, encounter outside forces that will allow them to bend, and the cytoskeleton just really helps it maintain while in motion. It interacts with motor proteins to produce motility and provides an anchorage for organelles. The three main components of the cytoskeleton are microtubules, microfilaments, and intermediate filaments. The microtubules are the largest structure, or the biggest in the in the cytoskeleton, and they're small, round, hollow tubes, and they're made of a protein called tubulin, and they're involved in the transport of cellular material, and they're very dynamic, so they can like switch shape. They function in cellular growth, vascular transport. As I said, they guide motor proteins using organelles, help move chromosomes, and mitosis and meiosis. The microfilaments are the smallest component of this cytoskeleton. They're composed of the actin protein, so they're referred to as actin filaments, and they're comprised of F-actin and G-actin. They twist like two pieces of thread almost. And the microfilaments are designed to resist tension. The microfilaments can be found just beneath the cell membrane, and some of them are anchored to the cell membrane. They play a role in cell motility, differentiation, division, and membrane organization. The last structure, the intermediate filaments, are like the middle-sized filaments compared to microfilaments and microtubules. They're made up of keratin, and it's a fibrous supercoiled protein. The intermediate filaments are like a meshwork that work together to serve like a net in the cell, and it helps protect them from mechanical stress. And it also helps the cell be able to bend without being broken. Cell motility is important in wound healing, and the cell senses the loss of epithelial integrity, which triggers cell motility and gene transcription. And it's important in embryonic development animal. Unregulated cell migration can be the cause for progression in cancer.
There are different types of signals in cell motility. Chemotaxis, haptotaxis, and durotaxis. Chemotaxis is the movement of an organism in response to a chemical signal. Um, Haptotaxis is directional motility or outgrowth of a cell, and durotaxis is when cells are guided by rigidity gradients, which arise from structural properties of the extracellular matrix. Uh, for this figure right here, it just has proteins associated with actin cytoskeleton regulation. And at the top, you can see the receptors. And then the next green box, you can see GEFs, which are guanine nucleotide exchange factors, um, which influence the proteins such as CDC42, RAC1, and RHOA. CDC42 activates WASP, which then goes on to activate ARP2/3, leading to actin regulation. CDC42 also regulates or also activates DIAP3 to also lead to actin regulation, while RAC1 can activate WAVE and also can activate ARP2/3, just like CDC42 can. RAC1 also activates PAK, which inhibits MLCK. It also,
You good? Keep going.
yo, like I can't see the PowerPoint.
Oh
Though.
yeah, damn.
What happened?
She
I'm
said she
guessing her
can't.
Wi-Fi went out again.
But pull the PowerPoint on your thing so you don't have to depend on her. Because I have mine up.
Son.
Man.
What the fuck?
Shit.
Some bullshit.
It really is. Damn. Oh.
So what? Like, I mean, I got my joint. I got it up now, but
Okay, so let her call. Let me see if she can call us back. Oh, shit.
I mean, just keep going, bro. We'll see if we can motherfucking finesse that shit out or something.
Fuck, I have this fucking homework to do too. Shit. Forgot about that. Damn.
alright, I'm about to keep going. No. PAK.
Is my music playing?
Hold up. Am I still going?
Yeah, just keep going, bro.
Alright. The PAK pathway activates LIMK, which inhibits coughlin, and that leads to actin regulation with inhibited coughlin. And then with the RHOA signal pathway, ROC can activate LIMK, which is inhibited and leads to coughlin inhibition, while also leading to the inhibition of MLCP that inhibits MLC, leading to actin regulation. The RAS family. RAS family is a small G protein, or RAS is a small G protein that acts as a molecular switch, transmitting cellular signals through effector proteins. Some of the well-known RAS members are HRAS, KRAS, and NRAS. RAS plays vital roles in control of cell proliferation, differentiation, and cell death. Uh, they also regulate these functions through gene expression. Uh, RAS switches operate. RAS switch operates by alternating between active GTP bound state and inactive GDP bound states. The extra phosphate on the GTP holds the switch region in a loaded spring configuration. Um, Upstream signals can stimulate dissociation of GDP from GDP bound form, and the reaction is stimulated by the regulator called GEP, also known as the guanine nucleotide releasing factor, and is regulated by an upstream signal. And our next figure is just a figure showing the RAS, the whole RAS like signal pathway, including CC42, RHO, and RAC. On the left side, you can see RAS activating CDC42 by RAGP1, and that leads to actin cytoskeleton regulation on the left side. Coming down the middle, you can see PI3K activating RAC, which activates PAK, going on to activate MEK, and activating JNK. And then on the branched off on the right side, you can see RAC also activating RHO, which leads to the formation of stress fibers and focal adhesions. It's on you, Keith.
All right, so cell migration is broken down into 4 different parts. The first step is the protrusion of the leading edge, which is followed by adhesion, which is new attachments that the cell creates, and then the cell contracts, which is the third step, by myosin, actin motor protein, and cause retraction, which is cell catching up to the rest of the body. In order to regulate cell polarity, membrane receptors must detect an asymmetric signal from outside the cell. Cell polarity requires the orientation and capture of microtubules at the lead edge. Receptors like RAS are small like G proteins, such as the Rho family. The Rho family is RAS-like proteins that regulate the morphology and polarity of cells. And they're also included in cytoskeletal changes and of the leading edge and at the trail edge. Protrusions are driven primarily by forces that are produced by actin polymerization. There are 2 main types of actin protrusions, which are present in motile cells. Lamellipodia, which are sheet-like protrusions, and Philopodia, which are finger-like protrusions. Lymphopedia are composed of branched networks of short-actin filaments, and filopedia are composed of long unbranched bundles of actin filaments. Formation of adhesion contacts. Cells bind to extracellular transmembrane receptors called integrins. Integrins are can indirectly cross-link the extracellular matrix to the actin network. Integrins are bound to the extracellular matrix to form clusters called focal adhesions. It links to the substrate and allows the cell to exert force and gain traction in the cell's motility. The amount of tension between the cytoskeleton and the extracellular matrix of how cells feel the rigidity of their substrate. Dehesion and retraction of the trail edge. Dehesion is when the cells contract due to myosin 2, which is an actin motor protein, which is activated by Rho, which exerts a force on the cell to allow it to be motile. When the force or tension in the cell is greater than the strength of the adhesion, the rear of the cell shortens elastically and contracts further.
Metastasis population of cells that have lost their normal controls of growth, and they spread into the lymphatics, the blood vessels, and cavities to form a second tumor. The primary site is where the malignant neoplasms or the cells that arrive there. The secondary site is known as where the metastasis has spread to. The metastatic cascade determines how tumors spread. The four main processes are detachment of the primary tumor, cell migration and invasion, intravasation, and extravasation. The detachment of primary tumor basically means that there is a cell migration deals with the tumor cells that enter the circulation. Intravasation is when the tumor cells enter into the blood vessels. The extravasation are the attachment of activated tumor cells that enter the blood vessels and exit from the blood vessels, then divide and begin their second tumor. All in all, cell migration is critical for our understanding of both basic biology and the pathology of diseases. And that will be our conclusion for today.